with the rest of the class. Now, like I said, we're going to cover the reproductive system in what we have left, which is about an hour and a half. And that's really not enough time to do everything I'd want to in the reproductive system, so I'm just going to cover a couple of things. First off, since at the end of the semester, I'm usually kind of rushing to get through this stuff, which actually doesn't happen every semester, but in the summer it always does. The lab material for the reproductive system concentrates on the male system, and the lecture material concentrates on the female system, which is to say that for the practical, the case that has to do with reproductive stuff is about the male reproductive system. And also, if you look through those cases, you'll notice one of them is obviously about the respiratory system, two of them are obviously about the digestive system, and then the fourth one is mostly about reproductive system, but the male reproductive system is tied in with the male urinary system. So it kind of, one case covers both of those things. If the case on the reproductive system was about the female system, then the urinary system wouldn't be that closely tied to it, but there's that. Now, we're in the last unit of the book, which is human development and the continuity of life. There are two chapters in here, but really we're just concentrating on this chapter as far as the assignments you have. I do want to go into the other chapter just a little bit to kind of set some things up here. Now, this chapter goes through kind of the whole thing, from fertilization to postnatal aspects of development. And I really don't want to go through all of that, but I do want to set up the reproductive system a little bit, in that the point of the reproductive system is fertilization, to bring the sperm and oocyte, which is the formal name for an egg cell, the Greek version of the name for an egg cell, together. And oh, I thought there was a picture in here. Maybe it's in the... No. Huh. I thought it was in the fertilization section of this picture, this chapter. Maybe it's not. Oh, here it is. This is a picture of what you would expect after the fertilized egg cell has divided. So this is actually two cells that are half of the two halves of the fertilized egg cell. The two other dots that you see in here are additional components of the results of the female reproductive cycle, which I'll talk about a little bit later on. But really,
what if you have twins? Would there be four of them?
So if you had twins, then you would be looking at two of these things. Right. If you're talking about identical twins, then those two things came from a full cleavage of the original fertilized egg cell. If you're looking at fraternal twins, then it's two separate fertilized egg cells. But considering what's enclosing this is within one zygote, one individual. A full cleavage would break this apart and you'd see two of these. So this is one division giving us two cells from the original fertilized egg cell. When those two cells divide, we have 4. When those 4 cells divide, we have 8. And then 8 divide to give 16, 32, 64, et cetera, et cetera. In the early series of divisions, which we see represented here, it's really just a cluster or a clump of cells. They eventually start to segregate out into what's depicted here called the blastocyst, where the clump of cells is off to one side, and then there's sort of a spherical appearance here. But the point is that all of the cells we see at any of these stages are really just from that original fertilized egg cell. When a cell divides, the two that we see here, for instance, those are just copies of the original cell, what we would call clones. Now, thanks to Hollywood, a lot of people think of clones as being, multiple copies of Arnold Schwarzenegger on the screen. But really, clone just means two cells or organisms or whatever it might be that have the same DNA. So every cell in your body is a clonal descendant of that fertilized egg cell that gave rise to you through the type of cellular division called mitosis, which starts with the DNA being copied and then half of it going into one cell and half of it going into the other cell, so that both cells have all of the original DNA of the first cell. With a few minor exceptions, all of the cells in your body have the exact same DNA in them. What makes them different is which parts of the DNA they use. So muscle cells use the genes for myosin and that sort of thing. Neurons use the genes for acetylcholine receptors or whatever they might need for their particular use. Skin cells use the gene for keratin. All of those genes are present in all of the cells of your body, with a few minor exceptions. And it's just a matter of how they express those genes that differentiates them in different types of cells. What's important about the reproductive system is that the cells that we're talking about here, the sperm and the egg cell, the oocyte or the ovum, they are made through a different type of division, which is called meiosis. Now, in mitosis, it's simply the DNA in the nucleus of a cell is copied, so there's basically twice as much DNA, and half of it goes into one cell and half of it goes into the other cell. And you have two identical copies of the original cell. In meiosis, it's a little bit more involved. There's a couple of divisions that take place, and the end result is, the end result is cells with single, how should I say this? Cells with half the DNA of the original cell, so that you can take two cells, each with half the DNA that you need, put them together and get the full DNA of a new person. I thought that there were pictures of this here. Well, this is the process specifically referring to sperm cell production. And let me make this picture bigger. There's a stem cell, which for sperm is called the spermatogonium. It divides through normal mitosis, giving rise to two cells. One of them, which has the arrow pointing back to the spermatogonium, stays as the stem cell, and the other one, called the primary spermatocyte, then goes through meiosis, which is 2 rounds of division. Now, inside each of these circles, they have a designation referring to the DNA, which is 2N, or it says 1N here. Most sources will just have N. N represents the number of unique chromosomes in a species genome. So 2N means 2 of each chromosome. 1N means 1 of each chromosome. And most sources, including this book, except for this picture right here, will just say N, because 1 times N is N. Okay, just a mathematical truism there. So it's kind of weird in this picture they have the 1 in here, because you really don't tend to see that. Now, how many chromosomes are there in the human genome? 46. That's how many chromosomes we'd find in most every cell in your body, but it's really because there's 23 pairs of chromosomes. So the N number for humans is 23. Now, 46 is a fine answer to that, because with the exception of sperm and egg cells, there are 46 chromosomes in all the cells. It's just you have one chromosome from your mother and one chromosome from your father for each of the 23 pairs to equal 46 total. Now, those 23 chromosomes are ordered or numbered based on their length. Chromosome number 1 is the longest, and chromosome 22 is actually 21 is the shortest. And then the 23rd pair are the sex chromosomes. If you're female, like most of the people in this room are, you have 2 X chromosomes. Men have 1 X chromosome and 1 Y chromosome. And that's what determines gender. It's because of genes located on the Y chromosome that make the fetus become male. Without the Y chromosome, the fetus becomes female, which is the default sort of setting, if you will. In making sperm or egg cells, the parents pass on one of each of their chromosomes. And so as far as the sex chromosomes are concerned, a mother always passes X chromosomes on because that's all she has. The father passes either an X or a Y chromosome on, and it's kind of a 50-50 split between which ones are passed on. If the resulting embryo gets a Y chromosome from dad, then genetically it's a male fetus. And if it gets an X chromosome from dad, it's genetically a female fetus, which makes Henry VIII's story kind of unusual, as he's killing his wives because they don't produce sons, but it's really because he wasn't passing Y chromosomes on. Okay. Actually, there's a little bit more to that. There's some evidence. If you look at the population, there's not a perfect 50-50 split between men and women, saying that there's more than just the XY chromosome thing to it. And there's evidence that it's also dependent on the resources available in the environment, which basically means the availability of food. When resources are plentiful, which in the modern age is not unusual, people are more likely to have female children. Okay, just reproductively beneficial if the resources are there to invest energy in making female children, because it's the best strategy to be sure that your grandchildren are definitely related to you. Okay. When resources are sparse, it's better to have male children because less energy is required for males to reproduce. And I apologize on behalf of my gender for that. But also there's subsequently less definite proof that your grandchildren are actually yours. So there's a little trade-off there. Also, I recently found a nice video talking about gender differences, which I tried to put on Blackboard, but there was a problem with YouTube and Blackboard integration recently, so I wasn't able to put there. But if you look on YouTube for sex determination or something like that, first off, be careful about what you watch. But somewhere out there, there's a pretty good video that talks about it. And it says that actually there tends to be many more male children born. Okay, and the suggestion from the video is that the male behavior patterns put those individuals at risk more often. So the more male children that are born, the more that will survive to pass on genes. Outside of that video, I don't know how true that is. I've not looked at the data, but that's their the hypothesis they're putting forth there. But it's an interesting story, and it also talks about the resource availability thing there. Anyways, so that was kind of an aside talking about what the N number is. So the stem cell, the spermatogonium, has 46 chromosomes in it. It duplicates all of that DNA and divides so that each of the resulting cells also has 46 chromosomes. Then one of those cells remains as a stem cell to do this again in the future. The other one becomes a primary spermatocyte, which then starts into meiotic division, so of which there are 2. In the first round, the DNA is actually copied again. So what is 46 chromosomes in the primary spermatocyte becomes 92 chromosomes. But then the chromosomes actually split up so that there's 46 chromosomes, but there's pairs of duplicates there. And half of them go into one cell and half of them go into the other cell. So these are said to be have 1N, or just 23 chromosomes, although they are in fact duplicated there. But it's like the maternal chromosome goes to one and the paternal chromosome goes to the other. And that just divvies up randomly among all 46 chromosomes. And then those duplicated chromosomes split again. They don't copy the DNA again for the last or the second division. They just take the already duplicated maternal chromosome 1, for instance, and one goes into this cell and one goes in the other cell. So we end up producing 4 individual cells. The general name for these cells that are made at the end of meiosis is gametes. When we're talking about making sperm cells, they're specifically called spermatids. Then in the testes, the spermatids undergo changes where they become spermatozoa, or the mature sperm cell. Sperm is really just a DNA delivery device. So they lose most of their cytoplasm, and all that's really left inside is the DNA. And then this kind of thick part behind the head of the sperm has a bunch of mitochondria in it, which make ATP, which is what mitochondria do, that powers the movement of the flagellum so that the thing can spin, move, whatever. Now, just a nod to the resource thing I was talking about, these mitochondria need energy. They need some sort of energy-rich molecule to make ATP from. In the ejaculate, in the fluid that's made in the male reproductive system, there's a high concentration of mainly fructose, not glucose, but fructose, which can also be used to make ATP to help power them. But once that ejaculate is delivered into the female reproductive system, it will dilute into the female mucosa, and the fructose that was there is not easily available to these cells. So they have to basically get their energy from molecules available in the female reproductive tract. If a woman is taking in proper nutrition, then her uterine mucosa is going to be rich in carbohydrates, primarily meant to help nourish the zygote before it implants and the placenta forms in the second trimester, or at the end of the first trimester. But the sperm, as they're moving through, can make use of those carbohydrates also. And so a woman who gets plenty of good food, well, nutritioned, that's not the right word. You know what I mean. Then she provides an environment that sperm can get more energy and therefore swim further. A sperm carrying an X chromosome is going to be slightly heavier than a sperm carrying a Y chromosome, which means it needs more energy to swim. So in a high resource environment where the woman's taking in nutrients, that'll make it possible for the heavier X carrying sperm to swim and therefore reach the egg. And that partly explains why female offspring are more common in resource-rich environments, which again damns Henry VIII a little bit further. You're the king, you've got lots of money, you feed the women in your court very well, so they're more likely to produce female offspring for that. Now the numbers, the study actually looked at this, the numbers are pretty slight. It's the difference between like 55 and 45%. It's not a huge increase in female reproductive, production of female offspring. But it does tip things a little bit in that direction. And it has something to do with what nutrients are available in the female reproductive tract. Okay. So that's essentially what meiosis is. I'm showing you the sperm one because that's the first one that's available, but also spermatogenesis, the process of making the sperm cells from the spermatogonium, is a very symmetrical process. One cell gives rise to 4 equal cells, which then undergo changes to become sperm. Oogenesis, the process of making egg cells, is very asymmetrical. And actually, let me go to that, a picture of, whoops, that real quick. Okay, so here's a comparable picture of the meiotic process in the female reproductive system. So it's fallen off the top of the screen a little bit, but up at the top is the stem cell, which is called an oogonium, and it divides. One of the cells is going to just cycle back and continue to be a sperm cell, a stem cell. The other cell will continue on. Now, there's a little quirk in this picture because they're putting these stoplights in, which I'll explain in a second. It says primary oocyte over here, but that's actually what you call this cell here. The other cell is not really a primary oocyte. It's a cell. It's still a spermatogonium. That again, oogonium. So the primary oocyte will then progress into meiosis. So we see one division, and then we see a second set of divisions. Now, the stoplights are showing you that the process of meiosis taking place in the female reproductive system starts early and waits until it's needed later on. So meiosis begins before birth in the fetal ovary. It'll get started, but then sit at the very first part of meiosis, called prophase, until after puberty. So from the time when a girl is born until she hits puberty, all of the cells that are capable of becoming egg cells have already started into meiosis, and they're just waiting for the hormonal signal for them to do that, which happens after puberty. Now, some of them will start right away, first cycle after puberty. Some of them will continue on. Some will sit and wait till they get a signal at the very end of the woman's reproductive years. So some of these cells that are present before birth aren't going to be completing meiosis until she's in her 4th or maybe even 5th decade. But that's what this dividing line here is getting at. That means that a girl is born with all of the cells that are going to become egg cells. That no new cells go through this initial aspect after birth. Okay, if you really kind of want to think about blowing your mind, when a woman gives birth to a daughter, she's also giving birth to all the egg cells that will be her grandchildren. Okay, they have a lot of development to do, but they are all present there in the fetal ovary. Then the second division takes place. The first division takes place finally after the reproductive signals after puberty come along. And we go from the 2N designation, meaning that it has 46 different chromosomes, to the N designation, meaning that now both of these each has only 23 chromosomes. But the circles that are drawn here are not quite the same size. And that's to point out that this is not a symmetrical division. Outside of the box here, what we have is called the first polar body. It's basically half of the nucleus after the division. That's about it. The other one is called the secondary oocyte. This is the other half of the nucleus and all of the cytoplasm. That cytoplasm is very important for the subsequent divisions that I started off showing you, the 2, 4, 6, 8, 16, those divisions. And all of that cytoplasm is necessary to be able to drive all of those divisions. Okay. So it's an asymmetrical division there. Okay. Then this secondary oocyte will go through the final division and produce, again, an asymmetrical pair of cells. It'll produce another polar body and the mature egg cell. Oocyte is the Greek word for egg cell. Ovum is the Latin word for egg cell. So more often we call this cell an ovum. Oocyte is usually what we call it when it's still in the process of dividing. Now we have another borderline here. Above this line is before the sperm reaches this egg cell. Below the line is after the sperm reaches it, which is to say the secondary oocyte does not complete the second division of meiosis unless the sperm is present. So unless fertilization is going to take place, the oocyte's never going to finish dividing. Okay. For any reproductive cycle that a woman goes through that does not include the sperm reaching the egg cell, then the oocyte just stays in that sort of mid-meiotic state, and that's resorbed or expelled within the menstrual flow. It's only when the sperm is present that the egg goes through the final division. And again, all of the cytoplasm goes into the ovum so that it has everything that it needs to go through the subsequent divisions of the zygote. In the picture I showed you, which I don't have up on the screen anymore, the two cell division stage, I pointed out two little dots also. Those are polar bodies. Okay. There doesn't necessarily have to be three polar bodies like is suggested in this picture. The first polar body might never go through another division, and the second polar body here might be present. So those two black dots might be these things we saw here, or they might be some other representation of the polar bodies. But the polar bodies, as far as anybody knows at this point, don't have any real role to play in the development of the egg or the zygote, outside of just being the extra DNA that's no longer needed after meiotic divisions. Okay. Someday in the future, it might be discovered that there's something associated with the polar bodies that's required for proper development of the new individual, but nothing's been discovered yet. But I would not be surprised to someday in the future learn that there is something to the polar bodies that nobody's discovered yet. So this is meiosis for the female system, and the point is to make just one really big cell with lots of cytoplasm, and then the rest of the gametes are just polar bodies that aren't really needed. Okay. So again, this is the egg production of eggs. This is production of sperm. Now, as far as the anatomy for this section is concerned, what I'm interested in is mostly thinking about the process of gametogenesis, whether it's spermatogenesis or oogenesis, where that takes place and how those cells travel through the reproductive tract to eventually meet up. So for the male system, gametogenesis, spermatogenesis, takes place in the testes, which I'll show you a little bit more detail of in a second. The sperm then move into the epididymis, which is this thing sort of sitting on top of the testis. Now the word epididymis actually literally in Greek means on top of the twin. So that's, the name is just saying it's a thing on top of one of the two things hanging outside of the male pelvis. Okay. At the bottom of the epididymis, the reproductive tract continues out as the ductus deferens. Now it's accurate to call this the ductus deferens, although most people think of it as the vas deferens. And there's a reason why it's not really the vas deferens. Vas means vessel. Ductus obviously means duct. Those two words in anatomical terms are very specific. Vessels, like blood vessels, have simple squamous epithelial walls. Ducts have different types of walls. Some ducts have simple cuboidal, some have simple columnar, some have stratified cuboidal or stratified columnar. The male reproductive tract is primarily made of pseudostratified epithelia. And so it's more properly called a duct. It's a ductus deferens. Now, a vasectomy is, that word literally means cutting out a vessel. And so before people were more accurately calling it the ductus deferens, a vasectomy was literally cutting out a piece of the, quote, vas deferens, interrupting the path that sperm can move along and therefore rendering the man sterile. Sperm cannot move on through the reproductive tract, and therefore he can't fertilize egg cells. But aside from vasectomies, the sperm will move along through the ductus deferens within the spermatic cord, which I showed you on that model there. And the spermatic cord meets up with the seminal vesicle. So this is the end of the spermatic cord, and this is a seminal vesicle. They come together here, and they merge into the ejaculatory duct, which passes through the prostate gland and meets up with the urethra. And that's where the urethra becomes part of the male reproductive tract. The seminal vesicle makes seminal fluid, along with secretions that come from the prostate gland. The seminal fluid is the fluid that the sperm are actually in, what can also be called the ejaculate. And that's what's going to move through the rest of the urethra until it leaves the male reproductive tract. Where the urethra leaves the prostate gland and passes through the external urethral sphincter here into the base of the penis, it's joined up with the duct from this gland right here called the bulbourethral gland. This secretes more stuff into the urethra. Actually, primarily the bulbourethral gland secretes material into the urethra prior to ejaculation. The urethra, of course, is also part of the urinary tract, which, and urine tends to have a slightly acidic pH, which is not conducive to sperm health. So the bulbourethral secretions, or the pre-ejaculate secretions, will tend to be a little bit basic to help neutralize the acidity of any urine that might be present in the urethra and help to kind of clean the urethra out before the ejaculate moves through it. In this picture, looking at it from the side, this kind of pinkish, spongy-looking tissue surrounding the urethra here is called the corpus spongiosum. It's part of the erectile tissue. It's actually a fairly minor part of the erectile tissue. It's just what's around the urethra itself. These small little square-looking things here that are adjacent to the corpus spongiosum are representing part of a corpus cavernosum. There are two corpora cavernosa, which is the way you pluralize that in Latin. On midline, we're not going to necessarily see the two of them, but in cross-section, you can see the two of them. And with the corpus spongiosum, the corpus cavernosum are responsible for making the penis erect. Now, I want to go through some of the finer pictures here to give you a little bit more detail about what's going on. The first one is showing you the scrotum, which contain the testes. The testes are extrapelvic. They're outside of the pelvis. And most sources will talk about that being because spermatogenesis has to take place at a slightly lower temperature than body temperature, which is true. However, that's not a complete explanation because there are some animals that have internal testes and they have to deal with the body temperature issue too. It seems very unusual to put such an important gland outside in a very vulnerable position. Any damage to the testes will mean that this particular person cannot then pass on his genes. He's the end of his particular reproductive line. But the best theory I have explaining it is that because of the movement at the hip joints, if they were internalized, the testes would be more likely to be damaged inside the body by the hip joint or movement of the legs than outside. And that seems to be true for a lot of different organisms that run. Organisms that don't run tend to have internal testes, like dolphins or, what's the other example? I forget. There's another good example of it. But organisms that do run, and lions, gazelles, squirrels, rats, whatever it might be, anything that has hip joints that can move fairly quickly are going to have external testes. And that seems to be because it's less likely to damage testes, having them exposed to knees and whatnot outside the pelvis, than to have them inside the pelvis and have to deal with movements associated with the hip joints. The testes are really just a bunch of tubes. The first person that ever performed a public dissection of a testis made quite an impression on people. Everybody back then, not really understanding the anatomy of the system, thought the testes was just a place where there are a bunch of, and back then they didn't understand the idea of cells. They thought there were just a bunch of little itty bitty human beings swimming around, waiting to move on and find a receptive womb and develop into a full-grown human being. That was before cells were understood and they knew what sperm and egg were. So that was the impression of how things work. It actually has a bit of a gender political side to it, because it suggests that the male reproductive system is responsible for making the new human, and the female reproductive system is just there for gestation, which just kind of goes with the politics of gender. In fact, what it is these cells is where spermatogenesis takes place. Those sperm move through these tubes and eventually into the epididymis, and then down into the ductus deferens down here. The picture I was showing you before with meiosis is also showing a cross-section of one of these tubes called a seminiferous tubule. Let me just enlarge that a little bit. There are two main cell types in the seminiferous tubules to point out. One is the Sertoli cell, which is also sometimes called the sustentacular cell, or I prefer the easier one, which is it's also referred to as a nurse cell. And those are the cells that are making up the epithelium of this wall. Okay, it's a pseudostratified epithelium. The Sertoli cells go from the basal membrane all the way out to the edge. All of the nuclei you see in here are this spermatocytes going through meiosis, which is what really gives this the pseudostratified appearance. But those are not epithelial cells. Those are the cells becoming sperm. The Sertoli cells are the actual epithelial cells in here. And then the other type is the Leydig cells, which are also called interstitial cells. Those are in the connective tissue adjacent to the seminiferous tubule epithelium, and they play a role in hormonal control of what's going on here. So Sertoli or nurse cells and Leydig or interstitial cells are the two major types of cells here, besides the cells that are going through spermatogenesis. So those are the seminiferous tubules. Here's a more detailed picture of the structure of a sperm, which I already talked about. The sperm then go into the epididymis and then into the ducts of the ductus deferens, and then finally to the seminal vesicles, and then through the prostate gland within the ejaculatory duct, and then eventually past where the bulbourethral glands attach to the urethra, and then out through the length of the penis. This picture shows a little bit more detail about what's happening in the corpus spongiosum and the corpora cavernosa. First off to the side here, we have the mid-sagittal view, and then next to it is sort of a cross-section of what this tissue would look like. And here we can see the corpus spongiosum down at the bottom. That's really kind of a minor part of the tissue here. And the corpora cavernosa, the two things off to the side here, take up a lot more of that space. There are blood vessels going through all the length of the penis, and in the flaccid penis, there's not much blood in the erectile tissue, but then in the erect penis, what's happened is the cavernosal arteries have dilated, allowing a lot of blood into the tissue, and then smaller blood vessels throughout the tissue also dilate, and all of that blood makes the tissue what's called turgid, meaning stiffer, and that leads to the erection. And something similar is happening in the corpus spongiosum, but not quite as much, because if it became too turgid, it would actually pinch the urethra and keep sperm from moving. But a similar process is happening there. Down below, they talk a little about nitric oxide, which is abbreviated NO. And this is the signal that actually causes dilation of these blood vessels that leads to the erection. Under normal circumstances, the parasympathetic system sends a signal to the erectile tissue, which causes the nitric oxide to be released and vasodilation to take place. A very popular class of drugs has come along which works on the same basic principle. Viagra and Cialis. I almost said Celebrex. Viagra and Cialis work on the same principle. They're nitrate drugs. They are precursors to making nitric oxide. And so any little bit of parasympathetic signal getting in there when you have a nitrate drug on hand will make the nitric oxide signal very strong and therefore cause the erection. Now, really, nitric oxide is just a vasodilator. If you watch a Viagra commercial or something like that, the fine print will say, do not take with other nitrates, which would mean Cialis also. If somebody's just really excited about having an erection, they might say, hey, if I take both drugs, it'll be great. Which would be one way to take it with other nitrates, which would be a really stupid thing to do. But nitrates also include drugs that are used to treat chest pain, like nitroglycerin. And they work on the same principle. It's just that they are vasodilators for coronary arteries instead of the cavernous arteries. And so if you're on nitrates for your heart and you take Viagra for erectile dysfunction, or just for fun, as the case may be, you run the risk of having a precipitous drop in blood pressure because of systemic vasodilation. So that's why you're not supposed to take the two together. I'm going to come back to this later on, but this is just showing how a couple of important hormones control the activity in the testes and especially spermatogenesis. I'll actually come back to this later on because it makes more sense after talking about the female system. So let's talk about the female system. The organs that make up the female system, kind of like the urinary system, I think are kind of straightforward. You have the two ovaries, which are the glands, analogous to the testes. This is where the ova are made, or I should say the oocytes are made. And then the fallopian tubes or uterine tubes, which is the tube that the oocyte or ovum moves through to get to the uterus. And then the uterus connects to the outside through the vagina or the birth canal. So really, as far as the basic structure, it's very similar to the urinary system. There's two organs where everything starts, and then two tubes that carry things from that organ to one singular organ where something takes place, and then there's one last organ that connects to the outside. Okay. Very, very general description of the layout of those things, but there is a little bit of symmetry there. Now, the book shows structure of the external female genitalia, showing a few things, which is not really the focus of what I want to talk about in this section, but I do want to mention I've found this quite fascinating. Only a few years ago was really good evidence for this presented. This structure we see here, which comes to a tip right there, is the clitoris. it actually extends inside the body and wraps around a lot of the structures in the female reproductive system. It's essentially the same kind of tissue as the corpora cavernosa in the penis. It's a really extensive system, but it's all internalized. All that's seen externally is the glands of the clitoris, which would be right there. And the information that was just recently published, like in the last few years, was actually showing pretty extensively what happens when the whole clitoral tissue becomes engorged, kind of like the same thing as an erection. And I think they had an MRI video showing what happens in internally. It was really pretty interesting, and it's something that was never really studied in much depth until just recently. And I like that story just because it shows that in anatomy and physiology there are things that are still being discovered. Okay, a couple of years ago was the first time anybody found evidence of this one little tendon in the knee that everybody knew had to be there, but nobody had found. There's always still information being discovered about anatomy, and the structure and function of the clitoris is something that's only recently been fully described, and they're still studying. So the ovaries are where oogenesis takes place, and I'll talk about the ovarian cycle in a little while. The uterine tubes, or fallopian tubes, are the tubes that conduct the egg cell through to the uterus. There's actually not a direct connection between the fallopian tube and the ovary. It's possible for an egg cell to be released from the ovary and not get into the uterine tube. If that happens without sperm being present, it's not really a big deal. The secondary oocyte that's released will just be resorbed by the pelvic wall. If sperm get through the female reproductive tract and fertilize an egg cell that's released ectopically, then it's possible that that zygote will sort of try to implant into the pelvic wall, which is one example of an ectopic pregnancy and very dangerous. Because if that zygote starts to grow in that space is not suited to grow a fetus, and so that can start causing lots of problems for the surrounding organs. That needs to take place in the uterus, which is meant for that process to take place. Okay. The egg cell will move through the fallopian or uterine tube to the uterus. Fertilization actually takes place in the uterine tube. So a fertilized egg cell will move along, or an unfertilized egg cell can move along to this point. The other example of ectopic pregnancy is when the fertilized egg cell implants into the wall of the uterine tube. The uterus is what's meant to expand with the growth of the fetus. Uterine tubes cannot expand. So if it implants there, unless it's caught very early and something's done about it, the zygote and then embryo, as it develops in that space, will basically rupture the fallopian tube or the uterine tube, causing all sorts of problems. Okay. So two examples of what ectopic pregnancy means. And then we get into the uterus. Now, the uterus is made up of layers of tissue, like a lot of organs that we've talked about this semester. The outermost part is called the perimetrium. It's kind of like a serosal membrane covering that, just like the peritoneum or pericardium or pleura. It's not exactly the same. It's not the same kind of double layered membrane with an enclosed space between it. It's not quite the same thing, but it's still an outer covering. Most of the wall of the uterus is smooth muscle tissue. There's actually 3 layers of smooth muscles. One layer, the center layer, is oriented one way, which is either circular or longitudinal. I can never remember which. And then the other 2 layers, sandwiching that one in the middle, are oriented the other way. So if the middle layer is circular fibers, the 2 outer layers are both longitudinal. If the middle layer is longitudinal, the 2 outer layers are both circular. And I can never remember to keep them straight. But very thick uterine wall called the myometrium. That's the smooth muscle that's contracting during birth. And then the inner layer is called the endometrium. If you think about other organs that we've talked about, the parametrium is kind of like the serosal layer of a lot of those organs. The myometrium is like the muscular layer of the bladder or the digestive tract or something like that. The endometrium is like the mucosa, submucosa part of digestive tract, or the mucosa of the urinary tract. It has two layers. There's what's called the functional layer and the basal layer, sometimes referred to in sort of Latinized terms as the functionalis and the basalis, but functional and basal layers. The basal layer pretty much stays the same all the time. The functional layer changes in thickness with the events of the uterine cycle, which I'll talk about a little bit later. I'm not going to zoom in on this picture, but it does show a micrograph of what the tissue kind of looks like. It's not a great micrograph, so it's not really worth zooming in on that. But there are two layers of the endometrium, and they play different roles in what's going on there. The cervix is the opening of the uterus that opens up into the vaginal canal. And then here we have the information about the reproductive cycles of the female system. Now I want to go back up here to where it's talking about the ovaries. I already showed you this picture here about oogenesis, the meiotic divisions that take place. Talked about that plenty. This series of pictures is showing something related to that. The cells that we see present here are, so there's oocytes, the egg cells that are part of this process here, oocytes. But then there are also other supporting cells that support the oocytes as they go through the ovarian cycle, the series of events that happen on a regular basis in the ovary. Now, the ovarian cycle and the uterine cycle, the two things that I'm going to stress here for the rest of the course, rest of the class, are locked together based on hormones that we'll talk about in a little while. And we're going to discuss this based on the average length of these cycles, which is 28 days. There is variability from person to person. It can certainly be shorter or longer than that. And except for Lee and me, I'm sure everybody has their own familiarity with their patient cycles. Anyway, so what I'm talking about here is basically what's happening in the ovary during the cycle, what we call the ovarian cycle. Before the cycle begins, in the ovarian cortex, the oocytes are found in little clusters, just one cell, but it's clustered by some supporting cells, which are called the primordial follicle. Now, what we're looking at here is how these follicles then progress through this cycle, so that we're going to stick with this follicle idea for the most part. The primordial follicle is basically those oocytes that I talked about a little bit ago that are present before birth. So this is these little nests of eggs. It's sometimes called an ovarian nest. Sorry, I should say that differently. An ovarian nest contains multiple primordial follicles, and they're essentially present from birth on. Every so often, they get a signal to go into the ovarian cycle. Okay, so what's represented in part one here, the primordial follicle, is always there. When the ovarian cycle starts, when the hormonal signals get started, some of the oocytes, a couple, few, half dozen, something like that, are going to respond to the hormonal signal, and they'll progress and they'll start to develop. The first stage of that development, once they're stimulated by the hormones, they become the primary follicle. So we have the oocyte that's surrounded by a layer of what are called granulosa cells. Now in the primordial follicle picture, they point at these things and call them granulosa cells, because those are the cells that become the ring of cells surrounding the oocyte there. But really, granulosa means grainy-looking cells, or granule-looking cells. And so to call them granulosa cells at the primordial follicle stage is actually not entirely accurate. It's the same cell, but technically they're precursors to the granulosa cells. So that when the follicle starts forming, the oocyte is surrounded by granulosa cells. It's not mentioned in this picture, it's pointed out in the next picture, but there are cells beyond the follicle that will play a part. Okay? After a little while, several days, I don't know exactly how long, the primary follicle, as it develops further, becomes a secondary follicle. It's kind of unfortunate in this particular picture of the secondary follicle, the way that they represent it. Now, this is obviously a drawing. They could have drawn it however they wanted to. And, but the drawings are probably based on images seen in a micrograph. Okay, so these are probably traced of actual cells. And it's most likely that under the microscope, you're going to see secondary follicles that look kind of like that. Okay? But really, what you should see is the oocyte that's in the middle of the follicle is attached to one side of the follicle. What makes it a secondary follicle is when the granulosa region, which is several cells thick at this point, starts to open up and gives rise to a little chamber or vacuole or empty space within the follicle. The oocyte is not floating around inside that like this picture kind of suggests. Really, there should be some granulosa cells that extend from the wall of the follicle and reach out and are still connected and holding the oocyte in place. But once we start to see something opening up in here, it's a secondary follicle. This is a rather progressed secondary follicle. The whole thing's kind of opened up. But if there's any little opening in the granulosa cells, it's officially a secondary follicle. Then that secondary follicle continues on to get larger and larger. And now we actually see that oocyte sticking off a little peninsula, if you will, of granulosa cells, which is what we should see here. It's just a difference in how they drew these particular pictures, and it's probably based on the specific micrograph images that they worked from. The book here is calling this the tertiary follicle, which is just going, primary for 1, secondary for 2, tertiary for 3. More commonly, this is referred to as the mature follicle, okay, because this is the follicle at the end of its progression. Okay. The space that opened up, giving rise to what we would call the secondary follicle, has become quite large. It's labeled as the antrum in this picture. And in this picture, as opposed to the secondary follicle picture, it looks like there's stuff in the antrum, and in fact there is. As the granulosa cells are pulling apart and giving rise to that space, some cellular debris breaks apart. There's some fluid in there, so there's stuff in there. It's not a completely empty space, which is kind of represented in that picture. That's the mature follicle. Now, what's not represented in this picture, and it's just a matter of where they happen to take the picture, the mature follicle should be right at the edge of the ovary, because what it's about to do is rupture and release the oocyte into the space beyond the ovary. And that's what's happening in the next picture. So the dark purple that we see here is the wall of the follicle, the granulosa cells that are still there within the ovary. And then where it's opened up, what's coming out is that fluid and cellular debris inside, as well as the oocyte that still has a little halo, if you will, of granulosa cells surrounding it, which is what we see here. Now, this picture is really kind of strange on purpose. Again, these are probably all drawn based on micrographs. Ovulation, which is what we're talking about here, rupturing of the mature follicle, is only going to happen in a living system. Now, you can take an ovary and look at it under the microscope. You can remove it from an organism, slice it up and look at it, and you'll probably see some representation of follicles at different stages, depending on exactly when you get it in the cycle. But the likelihood of being able to successfully get a micrograph slide of a rupturing follicle with ovulation taking place is probably impossible, because if you're trying to remove an ovary to look at it in a microscope, you have to, by definition, be working with expired tissue. And ovulation is only going to happen in a living system. So this picture is probably based on a micrograph taken of an ovary in vivo, in situ, in place, when the ovulation is taking place. And I've certainly seen such pictures elsewhere of it happening, in fact. So we don't see any real detail of the cellular structure here, and that's because we're not looking at it under the microscope the same way we do for all the rest. But that's what's happening at ovulation. As far as time is concerned, this is the halfway point in the cycle. So if we're talking about a 28-day cycle, ovulation is taking place on day 14. Okay. Now, as far as the pictures we're talking about, we're almost done with the pictures. And that's because the first half of the ovarian cycle, what we call the follicular phase, is the development of the follicles. And it ends with ovulation. After ovulation, the follicle remnants, what's left over of the ruptured follicle left behind, is called the corpus luteum. And it doesn't really do anything. So showing several pictures of that, so we've got 3 pictures in ovulation. If we had 3 pictures of the corpus luteum, it'd be very boring because it all look the same. It doesn't do anything. But the second half of the ovarian cycle is all this structure here. So remnants of the follicle. It's releasing hormones that direct what's happening for the rest of the cycle. It's just not physically doing anything that's very interesting to watch. We call that second half after ovulation the luteal phase. Okay? The first half of the ovarian cycle is the follicular phase. The second half is the luteal phase, just named after the structures that are most important for what's happening. This is a micrograph of a secondary follicle, and like I said, the picture that we're looking at here was probably drawn from a micrograph. I wish it had been drawn from this micrograph, because it does show that the oocyte is still actually touching the wall of the ventricle. And the developing antrum is really just a few different places that are sort of opening up. So this is an early secondary follicle. And the picture that we have up there is a late secondary follicle. But it sort of gives you an idea of the sort of range of what it can look like there. I started to say that there are cells outside of the follicle. They're not labeled in this picture. They are labeled in the next couple of pictures. They're called theca cells, or some sources will call them thecal cells. They're not part of the follicle themselves, but they are important to the hormonal actions that take place there. So I just want to point those out. So that's the ovarian cycle, essentially. The uterine cycle is not on that page. Before I get to the uterine cycle, I'm not crazy about the representation of, there we go. The representation of this is not the best way to do it, but each book has to sort of pick its own way to do this. This represents the hormones that regulate the ovarian cycle, and actually it's fixing the uterine cycle, which we'll talk about in a second. It seems to be cutting off a little bit at the top. Yeah. So that's the top of the screen, but there actually is a little bit more above that. and I'm not quite sure why it's not coming out. So for a second, let me just show you here. So the screen's cutting off right there. There's another box above it, is what I'm trying to point out. Okay. So, oh, nope. Oh, it's a stupid projector. Okay, so what you can see here, it says GnRH. That's an abbreviation for gonadotropin releasing hormone. The box that's at the top of the picture that's cut off says hypothalamus. GnRH is a hormone that's released from the hypothalamus. Its target is the anterior pituitary. There are specifically cells that have GnRH receptors on them, and those cells are responsible for making a class of hormone called gonadotropins. Okay, so gonadotropin releasing hormone tells its target cells to make gonadotropins. Gonadotropins are the two hormones abbreviated off to the side, FSH and LH. These two here. Referring to them by their initials is perfectly fine. I strongly suggest you do that. It keeps you from misspelling words and getting things wrong. But FSH stands for follicle stimulating hormone, which obviously stimulates follicles. It's the hormone that makes the follicles progress through the primary and secondary and onto their maturing stage. The LH stands for luteinizing hormone. The name luteinizing hormone really is referring to the fact that it's responsible for making the corpus luteum. But there's a bit more to what it does. So FSH and LH are the gonadotropins. They target the ovaries and stimulate the follicles to do what they're supposed to do. FSH primarily, when it's stimulating the follicles to go through their developmental process that we just saw, stimulates them to make an estrogen hormone. Now, estrogen is not a hormone. Estrogen is in the class of hormones. The actual hormone that's made is called estradiol. Estradiol will tell the endometrium of the uterus to do its thing, which I'll explain a little bit later. It also feeds back as a negative feedback signal to the anterior pituitary and the hypothalamus to shut down the release of FSH and LH. Now, it's important that you notice that I said the release of. I did not say shut down the production of. While estradiol is released during the follicular phase prior to ovulation, it's not telling the anterior pituitary to stop making FSH and LH. They're still making it. It's just not releasing it into the bloodstream. So it keeps FSH and LH levels low. That inhibition is concentration-based, which means that the concentration of estradiol, as it builds up, will act as that inhibitory signal to a point. Once the estradiol levels get very high, which is probably around day 12 or so, and that's represented in the next picture with this thick line as opposed to the thin line here. So the thick line means lots of estrogen. And some sources will actually say it has an effect directly on the anterior pituitary. This book says it's just affecting the hypothalamus. Really doesn't matter which story is told because the end result's the same, which is instead of being this negative feedback signal, it suddenly becomes a positive or excitatory signal, which causes the release of, as far as this book's concerned, a little burst of GnRH. Other sources will say that it directly tells the anterior pituitary. Doesn't really matter which way it does it. It'll eventually cause all of the FSH and LH that's been being produced but not released to be released. And notice for the arrows of FSH and LH here, the one for LH is thicker than the one for FSH. Not a very quantitative representation of this, but it is true that there's more LH being released here than FSH. But there's this little burst of those gonadotropin hormones being released there. The LH burst is huge. The FSH burst is kind of minimal, but technically both of them are being released. This is day 14. What this huge burst of LH does is it causes ovulation. And so luteinizing hormone, which is named for producing the corpus luteum, actually causes ovulation. And after ovulation, the remnants of the follicle become the corpus luteum. So indirectly, it causes the formation of the corpus luteum by causing ovulation. After ovulation, when we're in the luteal phase, the corpus luteum then starts to produce progesterone. Now, it actually is also still producing a little bit of estradiol, but progesterone is the more important part of what's going on here. Okay. Now, here we can see the hypothalamus part of the box here. Okay. So progesterone ends up being the negative feedback signal, which shuts down the hypothalamus anterior pituitary signals, helping to end that aspect of the cycles. Okay. Progesterone also has an influence over the endometrium, which will change what's happening in the uterine cycle, which I talked about before. So keep in mind, pre-ovulatory, during the follicular phase of the ovarian cycle, the ovaries are releasing estradiol that tell the endometrium to do something. And then after ovulation, the corpus luteum, which is the remnant of the follicle that was releasing estradiol before, is now releasing progesterone, which is then telling the endometrium to do something different. And so that's kind of sets up the uterine cycle. So, this figure addresses what's happening in the uterine cycle, and I want to talk about that, but it actually puts a lot of this stuff together. That's not it. That's not it. There we go. Okay. So up at the top, this is the first book I've seen say this type of stuff, and I don't really care too much, but I want to point out, because I did sort of say this. Here are primordial follicles, primary follicles, secondary follicles, and then we end up with a single tertiary or mature follicle. Okay? That's a lot of information they put up here instead of cramming it in on the timeline down here. But what this is saying is that multiple primordial follicles are signaled with each ovarian cycle to progress through the primary and secondary follicle stage, but only one of them reaches the mature level. Okay. So now this is not a perfect system. When this doesn't work properly and multiple follicles go all the way through, that will lead to multiple births, twins, triplets, quadruplets, octuplets, whatever it might be. Now, a lot of multiple births are actually because people are taking additional hormones for in vitro fertilization, and those hormones throw this process off quite a bit. And so additional follicles make it to the mature level. But it should only be one follicle that makes it to that point. Okay. And that's what we see here. So we have the mature follicle, and here's the little representation of ovulation that we saw before at day 14. And then in the second half, a luteal phase here, we have the corpus luteum. Now, this is a little bit of a misrepresentation. It seems to suggest that there's a steady decline in the size of the corpus luteum through the rest of the cycle. It's not really true. Corpus luteum persists for a good portion of it. Then at the end, it starts to degrade down into the corpus albicans. Now, corpus luteum actually means yellow body. So apparently the tissue that makes up the corpus luteum looks yellow. Corpus albicans means white body. Okay. Albican is kind of like albino, that word. And so what happens when the corpus luteum stops functioning and starts to degenerate, it becomes a more whitish tissue, which actually kind of looks like scar tissue. So after ovulations, the ovary will have what look like little scars left over of the subsequent ovulations. I've always wondered why there hasn't been a CSI episode or something like that where they can, try to establish the age of a victim based on how many ovulations are present. But then I thought about it, it's not really useful data because you'd have to know also how many times she's been pregnant. So an ovary with just a few ovulation scars is either a young woman or a Duggar. You just can't tell which. Sorry. So this progression here is a little bit incorrect. It's really just happening towards the end of this. But I wish they'd kind of stretch this picture out a little bit so that the 0 day for the uterine cycle lines up with the 0 day for the ovarian cycle. But 14 and 14 definitely line up. And then 28 and 28 should line up too. Okay. Because during that same 28-day cycle, the uterus is doing its own thing, which is essentially that the functional layer of the endometrium is going through changes. The estradiol that's released from the follicles during the follicular phase of the ovarian cycle causes the functional layer of the endometrium to start to grow thicker and thicker, and blood vessels to invest into that tissue. And that gives rise to what's called the proliferative phase of the uterine cycle. At day 14, after ovulation, the hormonal signals from the ovary changes from primarily estradiol to primarily progesterone. There's not really any progesterone pre-ovulation, it's all post-ovulation. And so the progesterone level, progesterone signal switches the uterine cycle from the proliferative phase to the secretory phase. The endometrium isn't probably quite fully grown up at that point, so it's going to keep proliferating. And there, in fact, is still a little bit of estradiol signal to cause that. But progesterone causes uterine glands to start secreting uterine mucus, which is, among other things, the secretion I mentioned earlier that contains the nutrient molecules that are necessary for survival of the zygote until it fully implants and eventually establishes the placental link. And also can provide some nutrients to sperm that might be trying to move through the female reproductive tract. So that's coming from the secretory phase. At the end of the luteal phase of the ovarian cycle, when the corpus albicans starts to degenerate away, the progesterone levels drop off and estradiol levels drop off. And the lack of those two signals will then end the secretory phase, and the functional layer of the endometrium will start to degenerate and separate off from the rest of the uterine lining. And so at the beginning of the next cycle, that's when all of that tissue is expelled as the menstrual flow. So really the first 5 or so days of the uterine cycle is referred to as menses, which is the menstrual flow, and the proliferative phase isn't really going to start up until after the tissue slept off from the previous cycle. But really the proliferative phase is what's happening in the uterus in keeping with the follicular phase of the ovaries, and secretory phase in the uterus goes with the luteal phase in the ovaries. And the menstrual portion is just really the remnants of the previous cycle leaving. Of course, if a fertilized egg cell successfully implants into the lining of the uterus, the secretory phase will continue on. The corpus luteum, which would normally degenerate away within the normal 28-day cycle, will not degenerate away. A signal will come from the uterus saying, hey, something has implanted here. And that will keep the corpus luteum present, and it'll continue to secrete progesterone for a couple or so months to keep the uterus in the secretory phase kind of state until the uterus starts to produce its own progesterone eventually, which I think is somewhere around the end of the first trimester. And so, of course, if that happens, there wouldn't be menstruation from the previous cycle, and we wouldn't start into a new cycle. Now, below these two representations of what's happening in the ovarian and the uterine cycles are a couple of graphs representing the hormone levels. The screen's still kind of cutting off here, but at the bottom, what is cutting off is just a label that's just saying days of the cycle. So again, we have 0, 7, 14, 21, 28 all lined up. This is showing the levels of the gonadotropins from the pituitary and this is showing the levels of hormones from the ovaries. So FSH and LH are being released from the pituitary because of gonadotropin-releasing hormone from the hypothalamus. And so those levels start to grow a little bit, but they stay fairly flat until finally we see a peak around day 12 to day 14. The FSH peak is not very significant. The LH peak is. Then after those peaks, both fall off and get down to their low levels until they finally start to grow back again to lead to the beginning of the next cycle there. While that's happening in the ovaries, we see this. So this line is estrogen, which really means estradiol. And then the other line, which is pretty much flat in the first half, is progesterone. Estradiol levels slowly increase through the follicular phase. And the levels from about day 5 or 7 up till about day 11 or 12 are going to suppress the release of the FSH and LH from the pituitary. Then when the levels get high around day 12, that's going to cause the burst of release of LH and FSH with it, although that's not as important. That's going to lead to ovulation. Okay? So these hormones get these hormones started, and these hormones feed back to keep these hormones from doing anything until this hormone eventually peaks, causing these hormones to peak. It's all kind of tied together. Okay. After ovulation, estradiol levels drop down fairly low. They come back up a little bit. The other representations we saw didn't really represent the low levels of estradiol being made by the corpus luteum, but it is present. More importantly is the progesterone. When the progesterone and estradiol levels fall off again, coming up on day 28, that's going to signal the endometrium to slough off, and that'll lead to the menstrual flow for at the beginning of the next cycle. Birth control works on manipulating this system for the large part. Birth control pills are usually progesterone and estradiol pills at levels similar to what's going on in the luteal phase of the ovary. Okay? So if you're taking pills with those hormones at those levels, your body's going to think that you're post-ovulatory, and therefore you will not ovulate. And then you stop taking the pills for a week, and the levels will drop off, which will cause menstruation. And then as soon as that's over, you go back on the pills, and suddenly you jump from the low levels here to the high levels here. And you just fake your body out, and you go to a post-ovulatory phase, and so you never ovulate. And that's the point behind birth control pills. There's actually nothing about the pills that requires that to be taking place on 28-day cycle. So there's a brand of pills called Seasonique, which you take the pills for 3 months, and then you stop taking them for a week, and you have menstrual flow. And then you take them again for 3 months. One of the side effects there is that doesn't necessarily mean there won't be spotting on a regular basis. I don't understand it very well. I don't have one of my own, a uterus that is. So I'm not quite sure, but there does seem to be something inherent in the uterus that it does tend to try to keep on that 28-day cycle despite what hormones are surging through your system. So while Seasonique sounds like this great idea that you only have 4 menstrual flows through a year, if you read the fine print closely, it's not really true. But again, I don't have a uterus. I don't have any experience with it myself. This is just what I know of the system. There is also patches that release hormones on different schedules, and I don't know all the finer points of those patches, but again, it doesn't necessarily have to be on a 28-day cycle. IUDs work by manipulating the system also. I said earlier, if there's implantation, then the secretory phase will continue on, the corpus luteum will continue on to support everything at the beginning of the pregnancy. So an IUD is basically simulating implantation. It's causing the uterus to think that there's implantation, and so the ovary stays in the luteal phase, the uterus stays in the secretory phase as long as there's something there. Just sort of manipulate that. Just the signals that feed back from the uterus to the ovary after implantation. And that's about as much as I know about that, but sort of locked in. Now, I did want to go back and look at, that's not it. Oh, I know why I was cutting stuff off, because I was zoomed, oops, in so much. There we go. Okay, see, I nope. Sorry, never mind. It's the projector. I keep thinking it's a computer. It's the projector that's doing it. But I was trying to go back to this. Now, this is the picture from the male reproductive system. It's slightly unfortunate in the female reproductive system part of the book. They don't actually have an anatomical representation of this. They just have those boxes that I was just showing you. But here's an anatomical representation of the hypothalamus. Here's an anatomical representation of the pituitary gland. And GnRH, not GnHR, it's just a typo in the picture here. GnRH is released from the hypothalamus and causes, through the hypophyseal portal system, FSH and LH release from the pituitary. That's happening in women. We just talked about it for the ovarian and uterine cycles. It's also happening in men. Okay. FSH stands for follicle stimulating hormone. LH stands for luteinizing hormone. But those hormones are active in the male system too. So there's no point in referring to them by their names because the names don't make any sense. But FSH and LH have effects in the male system. FSH targets Sertoli cells, or the nerve cells, as I pointed out earlier. And it helps them to stimulate spermatogenesis. LH targets the Leydig cells, or the interstitial cells. And it causes them to produce and release testosterone. Testosterone also targets Sertoli cells. So FSH and testosterone together are necessary for spermatogenesis to take place. But those are there. Now here they're showing that testosterone, just like estrogen, acts as a negative feedback signal to the pituitary and the hypothalamus. They add in here another hormone called inhibin that's also a negative feedback signal. Inhibin's present in the female system also, but there's enough going on in the female system. As the book's describing it, they just left the inhibin out, but it's there too. And inhibin obviously inhibits. It's the negative feedback signal. Okay. I want to, just to have a little bit of time, I want to show you something about these hormones. Let me see if I can find a good picture of them. Okay, so these are the chemical structures of testosterone and estradiol, the primary estrogen that we've been talking about. They are both steroid hormones, which means that they have these 4 rings in their structure that they get ultimately from cholesterol. They're made from cholesterol. And here we see testosterone has an oxygen double bonded to the ring here. Estradiol has an oxygen-hydrogen group bonded to that same position. Testosterone has a carbon with its hydrogens, which collectively is called a methyl group, sticking out of the ring there. There's no such thing sticking out of the ring in testosterone, in estradiol. There's one enzyme that makes a change from testosterone to estradiol. Okay. The other stuff up here is the same between the two. But these two things, the oxygen there and the methyl there, are changed because aromatase takes this ring that has 1 double bond there and single bonds the rest of the way around and makes it into a ring that has 3 double bonds, which is what chemists call an aromatic ring. So aromatase makes the aromatic ring. And in doing that, it has to change this oxygen, has to remove that methyl. Okay. That's the only difference between testosterone and estrogen, or estradiol. One, two, three, four, five. Yeah, it's 5 atoms different. Okay. Now aromatase removes, let's see, a hydrogen there, 3 hydrogens there, a carbon. Where are the oxygens coming from? Whatever. I'm not following the chemistry that closely, but there are only 5 atoms that are different between these 2 molecules. However, the 2 molecules are vastly different in their effects, okay, which is very obvious just if you put a man and a woman next to each other. Okay. All of those differences are really based on the differences between these 2 molecules. How can two similar molecules, such similar molecules as this, account for such vast differences in the genders separations? I'm asking this question at the end of the semester to tie back to something from the beginning of the semester. What's necessary for hormones to have any effect? There has to be a certain receptor. While the testosterone and estradiol molecules are very similar, the testosterone and estrogen receptors are very different. Testosterone will not bind to an estrogen receptor. Estradiol or the other estrogens will not bind to the testosterone receptor. And it's the hormone receptor complex that has the effect. Okay, so testosterone will only have its effects because it binds to the testosterone receptor. Estradiol, likewise. Now, I pointed out the thecal cells or theca cells earlier. Those cells are actually where this conversion takes place in the ovary. Okay. The follicle is actually making testosterone, but it doesn't get very far out of the follicle. All those cells surrounding it will convert testosterone into estradiol, and that's what's secreted from the ovary. I don't know if there's such a thing, but if there's some sort of disease state where the thecal cells don't function properly, then ovaries would be putting out testosterone. Testosterone is a precursor to estradiol. And it's just that one enzyme, aromatase, that makes the change. And if testosterone is being released from the ovaries, there is some little bit of testosterone receptor present in the female body. And so it would have a very different effect than estradiol does, having binding to the estrogen receptor. So I just wanted to end on that idea, kind of tie back from the end of the class here to something at the beginning. Granted, not all the way to the beginning, just the endocrine system stuff, but it ties into the regulation things. I think I've hit all of the important things I wanted to today. There is more to the information. You do need to read the book and that sort of thing, get into it a bit more. And there might be some questions that go beyond anything that I've talked about, asking you to do some more research. But you have that. Remember, next week we're going to actually start at 9 o'clock. For parking reasons, you might want to still show up at 8, but if you don't, it's fine. The door will be covered up and all opens up at 9. We'll get started. All we're doing, you'll probably be out of here by 11, if not earlier next week. Have a nice week.